Melanoma is a type of cancer that affects the skin. Melanoma is the fifth most prevalent type of cancer in the world, leading to an estimated 106,110 new cases diagnosed in 2021. Melanoma is typically treated with chemotherapy, radiation therapy, or surgery, which can all be very invasive, painful, and associated with a variety of different side effects. As such, the aim of this study was to research new novel cancer treatments using compounds that have fewer side effects and can be used in combination with a synthetic drug. I decided to study the compound sanguinarine, which is an extracted compound from the rhizome of the bloodroot plant. I studied the anticancer and sensitizing effects of this compound on A375 melanoma cells. These cells were tested with sanguinarine alone and in combination with the chemotherapy drug tamoxifen. The hypothesis I proposed was that the sanguinarine treatment alone, and specifically in combination with tamoxifen, will increase apoptosis and decrease survivin or BRCA5, which is an apoptosis inhibitor gene's expression in melanoma cells. This was completed by extracting sanguinarine from a bloodroot rhizome herbal extract using mixed mode, specifically reverse phase and cation exchange solid phase extraction. The extract was analyzed using the UPLC to determine concentration and percent yield. After the extraction was complete, A375 melanoma cells were treated with 1 micromolar sanguinarine, 1 micromolar tamoxifen, and a combined treatment of sanguinarine and tamoxifen. After treatment, the cytotoxicity of sanguinarine was analyzed using the MTT assay, apoptosis was analyzed using DAPI nuclear staining, and the expression of survivin was analyzed using RT-qPCR. The cytotoxicity of sanguinarine was determined using a one-way ANOVA and Dunnett's multiple comparisons test. Results showed that the higher sanguinarine concentrations suggested in the literature, 16 micromolar and 8 micromolar, resulted in the cell viability being reduced to nearly 0%. Other results showed that the experimental sanguinarine concentration of 1 micromolar reduced the cell viability to approximately 75%, tamoxifen to 90%, and most notably, the combined treatment to 60%. DAPI-stained cells were analyzed to show indications of apoptosis, most significantly in the sanguinarine and combined treated cells. The expression of survivin was analyzed using a one-way ANOVA and Tukey's multiple comparisons test. Results showed that the combined treatment reduced the fold change of survivin expression to 0.13, sanguinarine alone to 0.22, tamoxifen to 0.78, and the vehicle was at 1.00. Results showed that the combined treatment was the most successful at downregulating the target gene in melanoma cells. Overall, results showed that sanguinarine showed promise of reducing the cell viability, inducing apoptosis, and downregulating survivin expression in melanoma cancer cells. In addition to this, the obtained results are comparable to that of previous studies. For example, one study obtained results showing a reduction in cell viability to 60% with a 1.02 micromolar sanguinarine treatment. Another study showed that sanguinarine decreased the expression of survivin in a dose-dependent manner. In the future, I think that based on my results, sanguinarine shows promise as a potential natural treatment for melanoma. For this to occur, it would be necessary to confirm the results with a higher population number. Apoptosis should also be examined further using flow cytometry, for example. And studies should be completed to determine the effects of sanguinarine on normal cells if it is to be used as a treatment. Thank you.